Hello, everybody. Welcome to Media Lab tonight. I'm Lee now. I'm currently a PhD student on the third floor at Camping with you. This is today, my colleague today. And we're very honored to co-host this event together with the BioChange initiatives. We were contacted by Stefan about a month ago, and he mentioned this amazing idea of hosting a BioChange event in the Media Lab. And we got contacted because of one of the projects we did, which we will talk about later tonight. It is related to biology. But we got excited about this actual organization of the events because our passion of trying to reach out to the larger MIT community and also potentially the larger community within Boston, within the US and beyond the US to the world.
So it has been a big hot topic of engineering and design biology in the Media Lab. Lots of groups in the lab actually started designing the project, starting their own interpretation about how biology can intersect, for example, computer interfaces or semiconductor or architecture and so on. And actually, if any of you knows a little history about the Media Lab, that the founder of Ourobotics used to say that being digital, like how digital is important to the Media Lab. But now recently he actually stated that bio is the new digital. And so we hope that through these events, and then we can start a platform that foster a broad communication between departments, between researchers, faculty, and students, and think about how to situate biology in science, engineering, design, and art. Yeah. So let's move to.
So, bioChanges is researchers, industry professionals, and professional designers. And 2-3 years ago, we started working with myself, Dr. Dominic Sedgate, Dr. Laura Alonso, and Dr. Victor Mahita on this idea of building biodesign facilities in London. And at the time, no one really had much of an interest in doing it. So instead, we started working on different projects. So one of the projects was using this 3D bioprinter, which I will talk about later. But we also worked with Raymond McCauley in BioCurious, he's also the head of biotech in Singularity University now as well. And the idea was, at some point, to build up all of these communities and have international facilities across the world that could be linked to the biomaker spaces as well. So then in 2015, Dominic introduced me to Professor Peter Childs, here on the left, and then Professor Miles Pennington. And they brought me along to the Royal College of Art and Design to teach the innovation design engineers kind of biodesign concepts, or as Miles kind of coined the phrase, the demystify synthetic biology and bioengineering. They also wanted to meet with all the heads of department to actually build these facilities within the Royal College of Art and Imperial College London. And these are actually going ahead with Professor Oran Katz, who is a visiting professor in the RCA and is setting up one of the labs in the Royal College of Art, and the Advanced Hack Space at Imperial College is also setting up another facility, and hopefully they will be linked. In addition to that, we started working with NUI Galway, where Professor Charles Spillane wanted to set up a lab where they could allow designers and artists to come in and use their spaces. And around the same time was when I met Hiroshi. And when Hiroshi and the Media Lab presented at the Royal College of Art, it kind of sparked an interest outside of everyone that was in innovation design engineering. And so like fashion designers and textiles all wanted to learn more. So that was when we started the first BioChanges event, which was in the bar in the Royal College of Art. From there we moved to another bar in Imperial College, and from that bar we went to Sir Alexander Fleming Hall in Imperial College, and now the MIT Media Lab. So it's kind of progressed from bar to lecture hall, and we're still working on building these facilities, but also building upon the community internationally, so not just in London and Europe, to actually connect it over to the MIT Media Lab, because I worked with Charles Bracchia back in 2013 on this concept as well. And so from there, we're still building upon it, helping them set up these facilities, and also a lot of projects have kind of started. So there are a few projects at the moment. One is actually, they're all looking for collaborators in MIT or even here. One is actually exploring the acoustic properties of biomaterials for novel applications. Then there's a biofashion project, which is using, I guess, shape memory polymers and biomaterials for kind of responsive garments. And while we were setting up the BioDesign courses with Innovation Design Engineering, we were actually running these microfluidics workshops, which are also being run here through David Kong and Head to Go Almost Anything. So it's kind of very similarities between what we were doing on both sides of the pond. Another project, this is at Central St. Martin's. One student is actually trying to grow human leather. She's trying, she doesn't have the facilities to do it, but she's done kind of the basic concept behind it. And then another one has actually merged bacteria with graphene to kind of create a conductive skin. And that's similar enough to an IDE project where they've done something similar for bioelectronics. Philip from BioFutures wanted me to show this as well because there's such an interest in synthetic biology, that his device is like a bento box where you can kind of centrifuge and extract DNA, then use PCR and jet electrophoresis on something that is, at the size smaller than the laptop. And they're starting a Kickstarter in March. And Philip actually set up a BioFutures event space in London, which allows people to get trained on all these techniques as well. In addition to this week I was actually talking to a group in Peru about setting up these floating labs across the Amazon. So that's another project we're trying to work on with the Fab Lab as well, and also the network in Latin America. It'll be interesting to see how that comes along. These are all of the people that we have worked with so far. So the people that have either spoken at the events or partnered up with us, including the MIT Media Lab at the top. And it's kind of, we're all working together on the same goals to try and expand what can be done with biodesign innovation here and in London. And I will talk about the company which I'm co-founder on, which is Ourobotics. So Ourobotics is a company that was founded to kind of change the way bioprinting is at the moment and to kind of revolutionize it. The meaning behind Ouroboros is that it symbolizes the sense of kind of constantly recreating itself. It's existed since Egyptian times, and then you've got one of our design advisors there, Dr. Southgate, kind of representing this is that we can create this technology and work on these different materials or software, but we need to, constantly reinvent it, which is what we're doing with some of our academic partners. So Gemma started working on these kind of basic bioprinters around 2011, 2012, in her kitchen and at home. As you can see, they're pretty rough models. And I was doing the same thing at the time, but we kind of both saw, well, they work, but they're not that great. So this is one of the projects for the Rio Tinto Sports Innovation Challenge, which was headed up by Dominic Southgate. And we used bare conductive ink and the bioprinter, which is a basic bioprinter, to actually to print this conductive mat. And as you can see, it's a pretty basic machine, and they're quite easy to get a hold of. So this was done 3 years ago. And then since then, I've seen other companies kind of come out with the same concept. And so even though Jem and I have a more advanced machine, we kind of thought that we should release an open source 3D bioprinter. So Ranuk actually wrote the protocol for an open source 3D bioprinter, which is the basic one. Oksana and Sim helped design the case. And the idea behind this is that we're actually planning to use this as a model that we'll open source and let anyone use, because the parts cost around £200, £300, but also if people want to buy it, then maybe we'll run a Kickstarter, or since the head is so basic, we might actually get another novel extrusion head. But the idea is that it's to demonstrate that these bioprinters are some of them are quite basic machines, and they should be available to almost everyone, either biomaking communities or children or people in the education system. So at the same time as that, we had Gemma was kind of working on these early designs for multi-material retrievable bioprinters, and they function essentially like a scar arm. And so using these designs, she then took the company to Hacks. And so from January until May, kind of started to develop the company further. And so we were able to print in liquid and then just do regular 3D printing. Then in July, we started to build the lab, which is essentially a BioDesign facility. And by September, we had it up and running. And then, even though the slide's a bit of a mess, by January this year, we have this kind of novel retoolable multi-material bioprinter. The idea is that it can do 10 multi-materials. It's got a swappable extruder system. It can print cells, biomaterials, and shape memory alloys. It prints in air and liquid. It's completely upgradable, modular, and only 13,000. So there's a short video of it working. So it just functions like a robotic arm where it comes in and it can sense anything. In this case, it's sensing a syringe, so it's printing using the syringe. But in any case, that could be any tool head there, and it could be picking up that and functioning like a robotic arm. So as of last week, Gemma won an award for the most innovative startup in Europe. That was actually in Dublin. We got some money for AdWords from Google, and then we built this also kind of novel incubator enclosure, which we're still working on, but the idea is to have that as a separate attachment for kind of physiological work. This is the main team at the moment. It's Gemma on the left. And these are the kind of, again, the text is merged together, but this is, these are kind of our advisors, so they're all quite well established in their fields. So you've got Professor Alexander Safalian, who has actually created the world's first synthetic trachea, which is in the little picture there, and synthetic nose. Brendan Corcoran, who works on veterinary medicine. Jimmy Moore, who works on microfluidics and medical device design. Daniel Green, who has his company, but is also a principal fellow in entrepreneurship at the Imperial College. Lucy De Silva was a professor of tissue engineering. Kevin is a co-founder of the SENS Foundation, and Dr. Dominic Serke, who is one of the main people in BioChanges, is also a senior teaching fellow in design engineering. And then James, who's hanging around here as well, is a expert in regenerative biology. Bill Liao is a co-founder of IndieBio. Dr. Ravi Goliathan and Trevor Kerr are both lecturers as well. And so a lot of these people are our advisors, but we're also working with them on interesting projects that they're working on in their fields. And one of the most interesting at the moment is using kind of robotic sensors embedded within cartilage or bone. And so the idea is that the kind of future of bioprinting is what one could actually be. And then exactly the slide is turning into an absolute mess. But so I guess if you need to email me, just use the biochanges@gmail.com, because otherwise you're not going to read any of that. Thanks. And I guess we'll go through questions at the end, because we've got a few presentations to actually get through, and we'll take a break before Joe's kind of keynote talk. So Helen, where's the laptop? Thanks again for everyone for coming. And in terms of biodesign and bioart, the person that's about to speak is possibly the person that's probably most world renowned for doing this. Biodesign is kind of there's a lot of hype around it at the moment, but Joe Davis has been doing this since 1985, 86 maybe. And the idea is that he's going to give his talk on what is, it's a mix, I guess, of his current work and past work. And so it's people like Joe Davis, like Oran Katz, and a few others that have been doing this for so long, and that's the idea of, I guess, around BioChanges is, one, getting the communities together, having these events and trying to build facilities, but also recognizing the people that have done it, before we ever started to even attempt to do it. So I'll just hand the floor to Joe and take over.
I'm really not going to talk about what I've done in the past. There are a couple of slides about some past projects, but they're only there for continuity. This is all about what I'm currently engaged in. These are flowers that were grown in space recently on the space station, the zinnias, in case you didn't notice. But I'm going to talk about a very, very unusual, very odd sort of garden, not the kind we'd imagine. This garden appeared about 4.5 billion years ago, the first time. And as far as we know it, it was the early Archean eon, roughly about 3.5 billion years ago, that life left its first detectable impressions. These consist of bacterial microfossils. It was a time when the Earth's crust had just cooled enough so that continents and tectonic plates could form. The atmosphere, well, for most of the history of life, the garden had a very different atmosphere than the one we know. There was no oxygen. It consisted of CO2, ammonia, sulfur dioxide, methane, and hydrogen. The organism is thought to have evolved in this reducing atmosphere. Can you all hear me? I'm doing good, huh? So, even though they evolved in this poisonous atmosphere, because that atmosphere would be poisonous to all air-breathing creatures today, the action of those organisms in this poisonous envelope of gases changed it into an oxidizing and oxygenated atmosphere after billions and billions of years, hundreds of billions of years. Yeah, you can see this oxygen only appeared in the last curve of this long spiral that reaches back, like 6,000 million years. The most widely accepted chronology considers the Great Oxygenation Event, the GOE, to have occurred sometime about 2.5 billion years ago. And the organisms responsible for changing the atmosphere into an oxygenated atmosphere actually appeared several hundred million years before the oxygenation event. Cyanobacteria thought to have significantly contributed to the Great Oxygenation Event, to the change in atmosphere. Don't seem to have appeared until about 200 million years ago. It's interesting too that oxygen in atmosphere was created as the waste product of these original organisms, kind of the reverse, where we're filling the atmosphere now with CO2 as our waste product. But until about 0.85 billion years ago, the level of oxygen in atmosphere was nil, was 1 to 2% of, for most of the time, of the level of oxygen today. And this 1 to 2% was enough. It was, yeah. It was the causal agent of the most devastating biological disaster ever to hit the planet, because oxygen is toxic to obligate anaerobic organisms. All of the breathers of the previous atmosphere were duly dispatched. And it's interesting that current multicellular life isn't the first time multicellular life appeared. Apparently it's appeared at least 20 times before. These are fossils of extinct multicellular organisms that lived within a few million years of the oxygenation catastrophe. And oxygen in our atmosphere reached its peak at 35% sometime in the Cretaceous, only to spike down again at the KT extinction. So I'm an avid fossil collector. I have a big personal collection. I have this fascination with fossils. This is a half-billion-year-old cephalopod. It's on a stone this big that's sitting across the street in Thomas Schwartz's lab. They grew to like 20 feet longer, these pre-cephalopods. Anyway, why am I fascinated with fossils? It's because they have this ability to transport the imagination, to bring you into lost worlds. And maybe for that reason, there's also something terribly plaintive and poignant about fossils. Because they also represent the great bottlenecks in history when life simplified. It was the passage of, yes, genetically diverse into genetically simplified populations. And the great oxygenation event wasn't the only mass extinction that was preceded by many others. In the Ordovician, 96% of everything died. Then in the Devonian, 70%. Then in the Cretaceous, more than 70%. It's just each time life, the diversity of organisms, there was more diversity historically in the Ordovician 500 million years ago than there is now, and Earth never recovered its the diversity of its biome until almost recovering it in the Cretaceous, and then there was the KT extinction. Yeah. We lament the passage of knowledge, the beacon of learning at Al-Andalus, or the Library of Alexandria in Egypt, or the Library of Nalanda in India. Each one of these great tragic losses of all the treasures of culture and civilization reaching back thousands of years. All the book burnings and acts of iconoclasm, and all these are kind of trivial by comparison with the unspeakable biological disaster of the mass extinctions. There are hardly any numbers to describe the extent of the lost libraries of genes and proteins held in uncountable disappeared species and whole kingdoms of life. More than 99% of all species that ever existed on Earth are estimated to be extinct. I mean, we can only imagine what fields like synthetic biology could accomplish with this vast retinue of lost genes, even if a tiny percentage of them were recovered. Unfortunately, we don't have much left in the toolbox. Most life on Earth lives in saline environments, in oceans and seas, both on the continental shelves and in the benthic deeps, in deep hypersaline anoxic basins, and in deep ocean vents, terminal hypersaline environments, evaporating seawater ponds, lagoons, marine salterns, solar salterns, and the great salt lakes of the world. The Dead Sea and the Great Salt Lake, but there are many, many others. This one's caribou gases on the Caspian, and this one, yes, in Africa, etc., etc. There are many, many salt lakes in the world. And also very high temperature hot springs environments, hypersaline hot springs environments. So As it turns out, human beings and other air-breathing, freshwater-dependent organisms, why we're by far the latecomers. We are the extremophiles. It's like our self-centeredness kind of skews what we think of as extreme. So halophiles are extremely diverse. They take many forms. They can endure extreme temperatures and extreme aridity, persist, yes, in environments without any water or very little water, routine exposure to high UV at high altitudes in dry deserts. There's a deep lake, called Deep Lake. It's a hypersaline lake in Antarctica, in the Westford Hills in Antarctica. The temperatures range from 11 degrees C to minus 20 degrees C. It never freezes because it's so extremely saline. And it harbors various species of halophilic organisms. These are this is a map of all of the subglacial lakes, liquid lakes, which also have to be hypersaline in Antarctica. This is one that they tried to drill into. They stopped short 330 meters the last season. They never made it all the way there, but they will. Some halophiles are known to survive in high vacuum, radiation, and temperature extremes of the space environment. It's the Long Duration Exposure Facility that NASA flew in the 80s, and Expose-E, it's in this century, or there's actually several of those. There was an Expose-E and an Expose-R flights that went to the International Space Station, and organisms were exposed to the same radiation in space environments. And these are European experiments, the European Technology Exposure Facility, also on the space station, and BioPan, which were lofted by Soyuz rockets into orbit, and then with exposed, and then deorbited it with a thermal blanket, and some 6 of those experiments were undertaken. So I do have an interest in space, I have an interest in myself, which means that I need to know who I am, where I am, what I am. I mean, that's the measure of sanity, right? We have to know these things. So I had 2 projects to transmit messages for extraterrestrial intelligence. This was ironically much more about transmitting messages to ourselves. Nevertheless, these are the most powerful transmissions to sun-like stars ever conducted. The first one at 100 million watts for 20, 30 minutes. The second one for several hours at 2 million watts from Arecibo, Puerto Rico, in which we transmitted the gene for rubisco, the most abundant protein on Earth. The protein it's not very efficient, but it takes inorganic carbon from the atmosphere and turns it into food. We don't make rubisco, we human beings, but all of the complex carbon that we're made from, that we consist of, was produced at one time or another by this protein. There's a collaborator in the Rubisco Stars project actually in the house. But these are the things, this notion about the failures of radar transmission, the sort of shortcomings of radar transmission. It's subject to inverse square law, so the beam weakens, even if it's collimated, it weakens with distance. And you can send one signal if you can manage it. There are only a few radars on Earth bright enough to outshine the sun, so it's highly competitive access. And so you send one message for 3 minutes. Frank Drake's famous message from Arecibo was transmitted for 1 minute in 1974. to sun-like stars 25,000 light years away. Anyway, gotta survive. Radiation, temperature, space environment, vacuum, you gotta have, because the speed of light is still a slow boat in the context of our own galaxy. It's a 200 million light years across. So if we send a message, if mitochondrial Eve, our common ancestor, had managed to send a message to the edge of our own galaxy, and it was received, understood, and immediately replied to, that message wouldn't be back yet. And when it did arrive back, it would be a different species, right, than the species that regionally sent it. So bacteria, see, answer. I got into like coding all this different kinds of information into bacteria and made up these elegant, very clever strategies to keep mathematical parity with input data, but to be biochemically and biologically friendly with the organism. Activity I'm still engaged with, but see, I wanted to make biological spacecraft. That was the whole idea, because I could make enough of them overnight so that there would be one for every star in the sky. One problem with this is radiation resistance, because space is a pretty harsh radiation environment. There's cosmic and galactic sources as well as the sun. And radiation damages DNA extremely. Heavy ions, gamma rays, and X-rays all have their effects and blow up DNA. And DNA organisms have these swing shift repair shops that go into action when damage occurs, and depending on the efficiency of that repair shop. See, we need radiation resistance to explore the planets because of the gravity well. We will have to supply the planetary exploration with factories in space, and those will be synthetic biology factories, obviously, and these organisms will have to be radiation resistant. So I went to the nuclear reactor like in the 90s, in January 1994. I went to visit the nuclear reactor at MIT, and I had to bring my own incubator because the reactor primary coolant is 50 degrees C, and I wheeled it, and it had to be pre-contaminated. So I found one pre-contaminated with tritium because once I put reactor coolant in the incubator, it was going to become nuclear waste, and so I had to find one that was pre-contaminated, and I actually brought it across campus on a furniture dolly. With my friend now Professor Stefan Wolfo at University of Heidelberg, we went to the reactor, and these are from 35 millimeter slides that are fogged from the radiation environment in the reactor, and the coolant, the primary coolant at the time, was much hotter than it is now because there was actually fuel particles in the water, and so we made a standard microbiological assay over 20 different media for micrococcus and yeast and enterobacters and bacillus and etc. And we actually got growth in two of these media, a media called TCNZ, which is for micrococcus, and a media called PYC, which is for yeast. And although we didn't get micrococcus or yeast, we got a very strange organism. We got a species of a chromobacter predominated with numerous bacillus species, 5 bacillus species, and they're growing rapidly, and they were still radioactive, so we had to go in and re-inoculate with fresh media and re-inoculate until it was radiologically but not biologically dead, remove it from the reactor, and then Boston University Hospital Hospital Clinical Microbiology Laboratory helped us identify the organisms. But see, that was a long time ago, and you can only grow, of this many organisms that exist, you can only grow this many. The rest are called unculturable. And we didn't have, back in 1994, we didn't have metagenomic tools. We could only detect what we could grow. And so now we have something called 16S PCR, which is PCR is a, do I have to explain to anybody what PCR is?
Yeah. What's PCR?
It's a copy machine.
Good one.
See, to explain PCR adequately, you'd have to explain the structure of DNA and the melting temperature and hydrogen bonds versus covalent bonds and primers, and it's a copy machine. And you can select what part of the genome of anything you can search for a particular thing. So 16S searches for the genes that make one of the RNAs that comprise the ribosome, and this is conserved throughout nature, and the tiny differences are enough to be pretty specific about what family of organisms. And so these are my colleagues, some of them in the room, who were reactor project pals. We decided to go revisit these with contemporary tools. We're sort of. We're using 0.2 micron filters, and Connor Campuson and Emma Koval already tested them for us, so we could notify the reactor about what our volume rate use of. We're going to use 5 liters of reactor coolant. We had to get a 50-degree incubator because we're going to simultaneously do microbiology. We're going to grow what we can grow, and then we're going to look with metagenomics about what we can find. And Michael Napolitano is like he's a brilliant member of our lab at Harvard, and one of the cool things he's done is to collect this like library of species of stuff that he's found growing all over the lab, like probable contaminants, and among them is the radiation-resistant organism Deinococcus radiodurans. And so we have these like little pneumatic rabbits from the reactor. They'll let us put it in these tubes like old-fashioned bank tellers, and inside, if I can get it open, I have 4 Eppendorfs, so I can put 4 different samples, and this will shoot into the reactor core where we'll get 10 to the 13th neutrons per square centimeter per second, and an unquantifiable, thus far unquantifiable bombardment of gamma rays. So we've accumulated we're going to use MySeq after we do the after we do the RNA sequencing, this PCR to tell us which organisms, which family of organisms. We're also going to do shotgun sequencing, deep sequencing, genomic sequencing of whatever we find, and match with this guy. And we're also going to use axolotls, regenerative organisms. They're remarkably regenerative. They can chop them into little pieces and they'll recover. You can take away many of their major organs, parts of their brain. You can make excisional and incisional wounds that heal scarlessly. And they've been exposed to X-rays and UV in experiments in the past, but never to particle radiation. And so, one of my collaborators is James Monahan, who's back there, who's laboratory at Northeastern who's got an axolotl ranch, and we're going to expose 20 or so. We're going to take tissue before and after, expose 20 or so axolotls to varying exposure to 10 to the 9th neutrons per square centimeter per second out of a beam tube, and we're going to do transcriptomics to look for spikes in activity of proteins in hopefully in response to radiation. And we're also going to use tissue from the standard laboratory moose, and from an African spiny mouse, a newly investigated regenerative organism in our rabbits. They regenerate amazingly well. And do we do horrible things to them all the time? But 16S PCR and metagenomics can contribute to biological spacecraft in other ways too, besides finding genes for radiation resistance. And for that, we'll turn to the subject of salt again. We're calling halophiles. The oldest DNA, until recently, the oldest record for intact DNA is 700,000 years from a horse ancestor frozen in the tundra. George Church told me that I should note that it's pretty beaten up into like 100 base pair fragments. Also, we have sequenced the mammoth genome, of course, and we found insufficiently intact DNA for tens of centuries to identify. But see, unless they're found in these are other unusual circumstances, like frozen in the tundra, the chances of find readable DNA are extremely rare. There are various spontaneous processes, notably depurination and cytidine deamination, that are at least comparable to cosmic ray damage. Those are cosmic rays as seen in my cloud chambers, which I'm not going to explain right now. The exception to all this lies hidden in ordinary crystals of salt. See, when salt forms in brine, the salt crystals themselves expand and contract at a different rate than the brine itself, and so it ends up with these inclusions, these fluid inclusions in the crystal, and this has become a tool for geology. Note that Morton table salt, the salt that's on the street outside, is likely Permian or Devonian from mines in New York or Pennsylvania. That the only current salt we get is sea salt, and that's loaded with bacteria. So these are inclusions in Permian halites and in Devonian halites. So, there's a science that's traditional in geology. It's called fluid inclusion microthermometry, and it uses these inclusions in salt crystals and other crystals, calcite, gypsum. You take the crystal and you put it on a special stage, which is like a Peltier device in a PCR machine. It variously precisely heats and cools the crystal. And when you do that, this vapor bubble appears because of the low pressure the crystal is expanding or contracting, and this vapor bubble appears, and then you gradually heat up the crystal until the vapor bubble disappears, and that's the temperature at which the mother crystal formed, and this gives us, so the crystal itself gives us a kind of recipe for the ocean that it came from, but also it gives us the temperature of the ocean that it came from. So there's a kind of geomicropaleontology that's been largely ignored. It was first we claimed in the 1960s, in 1960 and 1963 respectively, that living organisms, that ancient fossil organisms were brought to life from these crystals. And this was considered controversial, that contamination issues were suspected. Yeah, they were controversial. So, but in the 1990s, things changed. Then we got PCR and the tools, the basic tools of genomics and metagenomics, and we found many organisms. These from crystals of salt in the Atacama Desert, and Halobacterium and Archaea. This was all done with 16S PCR, and these are from Permian Triassic salts in England, Germany, and Austria, from a shallow ocean that once covered Western Europe. One of these, a Halococcus, was cultured from several various geographical locations. A species of algae was also found, this Drualiella. We think that its byproducts may help keep these organisms alive as a carbon source in the tiny droplet of saline. And also glycerol, one of its components, would be probably metabolized by these organisms, and they shrink and they form these cyst-like nodules that were first discovered in these fluid inclusions. And so the decontamination is normally accomplished with sodium hydroxide and hydrochloric acid, washes and sometimes flamed. Then it's dissolved in a high salt media with either yeast extract for carbon or casein-derived amino acids, glycerol, etc. And the option to that is to use this micro-drilling apparatus to breach the inclusions. So here's a map of worldwide salt basins all over the world that march all the way from the Precambrian to the Oligocene to the Cenozoic, our sort of current epoch. Thanks to Alice Gelman, we've compiled a list of these. We're still working on compiling a list of these and their associated mines. And at first I was interested and requested samples of Ordovician salt from the Canning Basin in Australia from my friends at University of Western Australia, but they didn't have any in their collection, even though it's this huge, giant formation. And I had to go talk to the petroleum company there. But anyway, I don't have to do that anymore because their Ordovician formations, halite, evaporates in North America, in Canada and the U.S. And likewise for the Silurian and the Carboniferous, the Devonian and the Permian and the Triassic and the et cetera. And so we're doing an expedition, great Paleozoic evaporate expedition. Two of my colleagues from lab are joining me, and photographer Ashley Clark, and our kind of researcher Alice Gelman. And George says he thinks he can get it paid for our expedition, so maybe we get it like a Winnebago or a hot air balloon or something like that and go to a salt mine and come back with crystals until we get a sufficient collection. And there's benefits to evolutionary biology, hardly provable algorithms of evolutionary biology that predict the evolution of genes across geologic time. We can check those, and it would be useful anyway for evolutionary biology to track the mutations across those hundreds of millions of years. But also, there are insights for the search for extraterrestrial life. These are the seeps, the water seeps on Mars that have been recently discovered. They're called Linnea. And NASA has analyzed the spectra of the salts that, like, the water comes out and it's brine or wouldn't be water, and it immediately volatilizes in the near vacuum Martian atmosphere, and it leaves these salts. And the salts gave us spectroscopic information, spectroscopic data. It's rich in perchlorates, poisonous to most organisms, but for several halophilic organisms. And anyway, with that formula, we can build a model for the Oceanus Borealis, the 3.8 billion year old ocean on Mars. So, I want to build 10,000 ocean models. I'm going to titrate from the salt samples from the organisms that I'm growing from the Ourobotics and the Silurian and the Cambrian, and over the 10,000 or many more, move the formula to the recipe for the Martian ocean to see where the terrestrial extreme organisms, and we can give them, because they live in Antarctica, we can give them genes for cold resistance, we can give them genes so they can metabolize perchlorates, we can give them genes, hopefully, after April, we should be able to give them genes for radiation resistance. So we can push the survivability of the terrestrial organism toward the Martian end. The goal is, of course, this is a garden for astrobiologicals. We want to create a Martian simulant, a seed. So why is this art? It's because it's a garden, because I want to make flowers for the planet, for the planets. And I'm not suggesting that we immediately posit these modified organisms on other planets, even though they would embody one of the most enormous landscape architecture projects ever dreamed of. We would thoroughly investigate these locations for pre-existing life, but it won't hurt to have the toolbox to do the job when it comes time. So these organisms don't only come from salts, and they might exist on Mars, but they come from some other places in the solar system too. This meteorite fell on Monahans, Texas, in 2009, I think, something like that. And it was 40 feet from these kids who were playing basketball, who eventually got $23,000 for this piece of rock that fell out of the sky. But they found salt in this rock. These guys from Johnson Space Flight Center went right there as soon as they heard about it, and lo and behold, they found liquid water inclusions in the salt crystals in the Monahans meteorite, and they did no biological investigation. They did not do 16S-PCR, but they did analyze the iodine isotope content. Iodine 129 over time, like a clock, decays into xenon 129, and they found this water in this crystal to be 4.7 billion years old. That's older than the Earth. And there was another meteorite, similarly old, with salts, of the Zag inclusions, which landed in Morocco within the same few years. And both of these meteors, and some 70% of the meteors that hit the Earth, are supposedly from this asteroid called Hebe, like Hebe-Jeebies. She's the goddess of youth. She's in this really long orbit. Yeah, doesn't come by very often, but what she leaves, what falls off, hits the Earth. It's in this perfect resonance to like transfer to Earth in Earth orbit. And Hebe. Wouldn't it be amazing now? Because see, Hebe and there are other planetesimals and planet-sized objects in the early solar system that rammed into each other, that delivered, that where there could well have been liquid water that delivered materials back and forth. And, so wherever Hebe's water came from, there are other salts in the solar system, we think. This one being Enceladus, which has an internal, oops. One more thing I wanted to say. Oops. Yes. Wouldn't it be amazing, see, if we find living materials in these salts from like the primordial planetary nebula, that would change everything. That's amazing, and that's something we should definitely search for. And there are other exophiles to be found, possibly on the ice planets of Enceladus. It has a liquid ocean under it, and Ganymede, Europa. So, that's it. And I want to thank all these people. Tom Schwartz, my MIT PI, Tom Borg, the reactor utilization officer, Andreas Merchant, Center for Bits and Atoms. Thank you very much. And James Monahan, Professor James Monahan from Northeastern, and Ashley Seifert of Kentucky, Alice Gilman for excellent research, and Connor Campbellson and Emma Kova for piloting the reactor filtration, and Michael Napolitano, not only for his contribution for the reactor work, but to other projects, and Seth Shipman and Jeff Naval, they're like my gurus, they're my benchmates, they're always making sure I don't blow up the world. And John Scheinman, also at Harvard Bees, Paul Reginato, who's here, participated in the reactor project, and Stefan Wolfel at University of Heidelberg, Ashley Clark, who's a photographer on the expedition, pending expedition, Tara Giannoulis, who helped pilot the reactor project back in 2011. We started to get way through all the bureaucracy about it and solve some of the problems, and because we solved some of those problems, we were able to like go faster now. Michael Sismore, who all participated, and then Church Lab in Harvard Genetics, and Thomas Schwartz in MIT Biology, who put up with me for many years, and MIT Nuclear Reactor Lab, and the Reese Institute. And thanks a lot. And BioChanges. Questions?
So from the salts,
Yeah.
you've been able to actually bring back to life,
They have.
And
And some of the
replicate-
organisms they brought back to life have never been seen before.
and they
That
replicate.
doesn't mean that they don't exist. They just haven't been discovered yet, or they're extinct.
And how far back? Earliest?
Ordovician, that's 500 million years.
500 million years.
And I think we can go further.
Sorry, I think this question doesn't make any sense, but I've been thinking about this for a while. Why do we want to bring them back? I mean, isn't the whole reason they die because it doesn't fit? Like,
Did you hear what I said about the toolbox, dude? I mean, we need those tools.
They're the
you
old
mean,
books.
you mean the species you bring back as a tool, or you're talking about?
They have proteins and genes that we can use that go directly into the toolbox. unless you want to be really evangelical about it and say, I mean, what doesn't belong? Because we destroyed it, or because some comet destroyed it, or because some accident of atmospheric concentration changed the Earth into an ice planet and further decimated the population. But all of these things were total accidents. It's like it's natural. Sure, it's natural, but so's Moby Dick and hurricanes and tidal waves and tsunamis. And I mean, if you look up art in the dictionary, it says opposed to nature, and for very good reason. Any other questions? I'll be kind.
Any of these organisms and these salt inclusions, do they have modern analogs? Are there living relatives so we can build a biologically?
Yes, yes, they do, but they're by and large genotypically and phenotypically distant than the existing halophytes that they're most closely related to.
So the halophiles in existence
They're both.
now.
This is only several experiments have been undertaken. A very handful of experiments, and mostly isolated formations, whatever's nearby the lab. No real surveys have been undertaken yet.
The halocytes?
Halophiles.
Halophiles. Where do those occur?
They occur in all those places that I showed you. That in hot springs, in deep anoxic hypersaline basins under the ocean.
They're
in
anoxic.
salt lakes, in
They're sort of
lagoons and
theory-
salt pans, and
Yeah. They're extremophiles in the terms.
Well,
in that
except
sense.
that most organisms on Earth live in saline environments,
Yeah.
are actually
So these are-
halophilic.
So they're the name is
That
from-
we
they're
are,
so-
we air-breathing, freshwater-loving creatures, we are the extremophiles. It's just our
Right.
self-centered point of view.
Right.
What's in your wallet?
But
Yep.
So the bacteria that survived the radiation, was there anything specific in the repair?
We didn't have I mean, genomics was just coming, it was 1994. We're going to answer those questions now. Our first experiments will be undertaken in April, we think. That's our plan.
So you didn't freeze them?
Pardon?
You didn't freeze them at the time? Come on.
You know how many times, in my minus 80 unthawed, actually the samples may be in the freezer at Boston University Hospital somewhere, but I doubt it. But we documented it carefully, and we have all of that documentation, and it was actually published in the Süddeutsche Zeitung in the regular newspaper in Munich. There was a story published about it. Yes?
I heard you made an apple of knowledge. Is that true?
That's true. It's a project that they have new news on today. In fact, it's an international collaboration. So we've sequenced the genome of this most ancient apple called Malus cerevisiae from Kazakhstan. The genetic evidence shows it is the ancestor of Malus domestica, all domestic apples, and the transcriptome, and we've compressed Wikipedia into first the top 50% most visited pages of Wikipedia into 100 megabytes. But my collaborators in Israel, Joshua Spitzberg, has just announced this morning that he's got down to 37. That's pretty cool. Paul Reginato also slightly assisted in that compression. So we're worried about error correction right now. It's so precisely compressed that it's like DNA manifolds. I have another option for further compression after I get the DNA form. But then again, it's like if you make one error, then all of the decoding falls apart underneath it. So we need to put error correction. We need to put error correction bases in the DNA chain at maybe 1/1,000. We want to beat the natural error rate, naturally occurring error rate of standard laboratory techniques and radiation. And to do that, we're thinking one additional base per 1,000. And then we're going to chip synthesize and grow polyclonal calyces. and each one of the polyclonal calluses would contain part of the total information, and I have collaborated with the USDA, and now we have we grafted Malus species onto disease-resistant dwarfing rootstock, which will give me trees that never get over 8 to 12 feet tall that I can keep in containment in special terraria, refrigerated terraria, because they're temperate plants. And to these trees, macro bonsais, we'll graft the shoots from our polyclonal callus. And if you have another half hour, I can explain our construction strategy.
Can you just clarify, you said Wikipedia, like taking the digital information equivalent of Wikipedia and encoding it in DNA?
Not all of Wikipedia, only
No,
the
but a chunk of it,
50% most visited pages over 4 years,
Yeah.
which is really ridiculous. You know, I criticize the search for extraterrestrial intelligence, these people who made these messages that made us all out to be like nice people with scientific peace-loving attitudes, and all terrible lies, and all the censorship, and so, what is knowledge? See, that's kind of part of this project. It's like, because everything is the reverse of what it should be, you know, and the reason it's should be forbidden is because it includes things like anal sex and human penis size and Justin Bieber.
What is the idea?
I won't censor. You will not be protected by censorship by Joe Davis, I promise.
Please don't. But the idea is to essentially encode the information into a DNA that then replicates and stays alive?
Yeah.
Is that the idea? Okay.
Inside the apple.
In the apple.
In the genome of the apple.
In the genome of the apple, but it goes on in time.
We're lucky that the
replicates
lifespan of the apple tree is 150 years, so the mutation rate is, that's a good thing, then once every 20 minutes.
Why you chose apple?
Well, for obviously poetic reasons, like where are you going to put, this it's sort of like, I spent 25 years putting all different kinds of information in the genomes of various organisms into E. coli and the eyes of flies and the ears of mice, and I realized that all information will eventually be contained in this archive because it fixes itself, because you can make hundreds of billions of copies overnight because we're just unraveling these amazing operations. I mean, this is the kind of development, this increase of operational increase, and the information handling capacity of DNA is increasing at such a rate that it's fair to say that we should think about this kind of technology replacing the internet eventually. Yes, sir. Yes, ma'am. Yes, sir. His turn.
Yeah, I already.
Would you say that the best place to store the human genome would be space or deep underwater?
You'd probably have a better radiation protector in the water, but depending on how deep you go, because see, the Earth itself is a radiation shield. So, like when you go high, you get more radiation. Probably when you go deep, you get, because there's less Earth on the other side of you, and you're closer to the core. I would have to say that if you wanted to, I mean, you know, the idea is to get, you know, to send out, to contaminate the whole universe, right? To bring the universe to life, right? To solve the Fermi paradox, that's the whole, that's what art has always wanted to do, to bring things to life, to change this kind of living material into this other kind of living material, or this animate object into an inanimate object, or vice versa. This is the dream. And remember, all those legends, all those stories came from the foundation of civilization, when we had first domesticated plants, when we altered, and livestock, when we altered the natural world. We realized we had the power, and so we wrote stories about, you know, Midas and Medusa and Daphne turning into a tree, and Moses throwing down his staff and turning it into a snake, and all that stuff. And Lot's wife into salt, and the whole transanimation of the universe. It's the age-old dream. And like the most exciting idea about animation, this trans animation is the search for extraterrestrial intelligence, because all we're doing is asking a question, and by asking a question, we're trying to bring the whole universe to life. It's a cool thing. Yes, sir.
So when you're sending out in the future flowers for planets, are you going to put in a little signature and you sign it?
Oh, no.
Sign
No.
it? No? Will there be any? No? Little nod?
I am not one of the top 50 most visited pages on Who cares? Dust to dust, dude. You have one life. You have one chance to do something that's really amazing, to change the world, to make things, dreams come true, right? You just go for it, right? You take up their dreams and you weave them into fine fabric. That's what you do.
I was thinking about when you were saying about this desire of human to make life, to animate things. So how do you think about robotics then? What's your opinion on that?
You're talking to a person who's like got significant in stainless steel.